In flowering plants, pollination is an important mechanism that aids in transferring the pollen released from the anther to the stigma of the flower. If the pollen is of the right type, the stigma induces it to germinate and produce a pollen tube. The pollen tube, containing the two male gametes, then secretes hydrolytic enzymes which help the pollen tube to digest the tissues of the style as it travels down to enter the ovule through the micropyle, a minute opening in the ovule. Inside the ovule, the pollen tube encounters the embryo sac, the female gametophyte, containing 3 antipetals, 2 synergids, an egg cell, and a central cell with 2 polar nuclei. The pollen tube now enters one of the synergids through the filiform apparatus, during which it ruptures and releases the male gametes into the synergid's cytoplasm. Of the 2 male gametes, one enters the egg cell and fuses with its nucleus. This fusion, known as syngamy or first fertilization, results in the formation of a diploid zygote. Meanwhile, the other male gamete moves towards the 2 polar nuclei and fertilizes them to produce a triploid primary endosperm nucleus or PEN. Incidentally, this fusion is known as triple fusion, as it involves the fusion of 3 haploid nuclei. After the triple fusion, the central cell is called the primary endosperm cell. Did you know that syngamy and triple fusion together represent double fertilization? A phenomenon unique to flowering plants. Double fertilization is followed by several post-fertilization events which eventually transform the ovule into a seed and the ovary into a fruit. These events include degeneration of the antipetals, synergids, and the development of the zygote into an embryo and the primary endosperm cell into the endosperm by repeated mitotic divisions. Endospermic development is of 3 types: nuclear endospermic development, cellular endospermic development, and halobial endospermic development. In nuclear endospermic development, which is the most common type of endosperm development, the primary endosperm nucleus divides mitotically and forms free nuclei. This division is sometimes followed by cytoplasmic divisions from the periphery. In the case of cellular endospermic development, which is seen in plants such as Utricularia, the division of the primary endosperm nucleus is immediately followed by cytoplasmic divisions. Halobial endospermic development, on the other hand, is an intermediate type of development where half the cells develop in cellular mode and the other half undergo free nuclear divisions. Interestingly, the number of free nuclei formed prior and post-cellularization differs greatly. Take the case of a tender coconut fruit, where the coconut water that you see is actually thousands of free nuclei. As the coconut matures, cellularization leads to a decrease in the quantity of water and the formation of the white kernel, the cellular endosperm. Therefore, in the coconut, we find both free nuclear endosperm made up of thousands of nuclei and cellular endosperm. Histological studies have shown that endosperm contains stored food in the form of starch or proteins, as in cereals, or oil, as in castor or coconut. This food provides nourishment to the zygote while it is developing into an embryo. It is interesting to note that the endosperm also forms a major source of nutrition for human beings. Incidentally, the development of the embryo, also known as embryogeny, occurs only after a certain amount of the endosperm has developed. In fact, in some plants, such as groundnut that belongs to the bean family, the mature seeds lack endosperm, as it gets completely absorbed by the developing embryo. When these seeds germinate, the cotyledons or seed leaves become the source of nutrition for the embryo. However, in mature seeds of castor, we see that the endosperm is retained as a storage tissue. In the case of cereals, the alurone layer, which forms a part of the endosperm, stores food as well as secretes amylase enzyme for the digestion of stored starch during germination of the seed. Studies by botanists have revealed that during early embryogeny, the zygote divides into two cells: the basal suspensor cell towards the micropylar end and a terminal embryo cell towards the chalazial end. The basal suspensor cell further undergoes mitotic divisions and forms 4 to 8 cells, which push the terminal embryo cell into the tissue of the endosperm to allow it to derive nourishment. Moreover, the terminal embryo cell also undergoes divisions to form a group of cells known as proembryo, which subsequently develops into a globular, heart-shaped and torpedo-shaped mature embryo. The structure of a mature dicotyledonous embryo is nonetheless different from that of a monocotyledonous embryo. A dicotyledonous embryo, for instance, consists of an embryonal axis and 2 cotyledons. The epicotyl, the portion of the embryonal axis above the level of the cotyledons, terminates with the plumule or stem tip. Below the level of the cotyledons is a cylindrical portion called the hypocotyl that terminates at the radicle or root tip, which is covered with the root cap. On the other hand, a monocotyledonous embryo, commonly found in the grass family, has only one cotyledon called a scutellum, situated towards the lateral side of the embryonal axis. The upper portion of this axis, known as the epicotyl, lies above the attachment of the scutellum. The epicotyl has a shoot apex and a few leaf primordia enclosed in the coleoptile, a hollow foliar structure. The lower end of the embryonal axis has the radicle and root cap covered in an undifferentiated sheath called the coleorhiza. Interestingly, the various parts of the embryo develop differently as the seed germinates. In this manner, The embryo, as well as endosperm, develops from the zygote and primary endosperm cell after fertilization has taken place in flowering plants.